beginning to recede, notice that there is very little vegetation in the water in this black and white image, and it's a little grainy, but it doesn't look like it's really there is much vegetation in that water line. Fast forward to 2011, when water levels are now at their near their recent minimum. If Phragmites has expanded to where it really make these kind of like these walls of Phragmites that we're used to seeing around the Saginaw Bay and other shallow water areas, like in Western Lake Erie or in Lake St. Clair or even the Green Bay area in Wisconsin, where Phragmites is really just taking advantage of that exposed lake bottom that may be nutrient rich and had very little vegetation and just created this really dense monoculture. If we zoom forward even further into 2018, this is as water levels were going up significantly. They're kind of near their recent maximum. And you'll notice in the top portion that where Phragmites has persisted, it's breaking up some. There's some natural drowning occurring. There's some stress on those plants. And while the water levels have gone up significantly and stressed the plant, it is persisting, and we still have Phragmites present. In that lower box, you'll notice this is an area where landowners actually used chemical herbicides to treat Phragmites. And so they were able to potentially take advantage of some of that stress that the plants were enduring due to that high water to get a really effective control. But in those areas where Phragmites has persisted, we're looking for new ways to control the plant that maybe wouldn't use herbicides. And one of those strategies may be cut to drown. So thinking about the future and even kind of the present about Great Lakes water levels, you may be thinking, why are we talking about cut to drown now? Water levels have been going down for the past 2 years. They reached their maximum in 2020, but they're going down and maybe actually have their average water levels this year. And the reason is this, thinking about climate change and the way that water levels are going to react to the changing climate is really unknown. And some of the most recent modeling exercises that look at how Great Lakes water levels are going to respond to climate change show that we're likely to get more extreme highs and lows in the future. So while cut to drown strategy may not be something that is going to be useful in this year or the next if water levels start going down, in these coming years, as water levels probably go back up, it's something that's going to be really important, a tool to have in the toolbox for managers. If you'd like to get a more of this historical perspective on Phragmites expansion, I'd encourage you to take a look at this geonarrative that we created a couple of years ago at this website. It's a really great resource for looking ahead, and also has kind of a tool where you can look at your area of interest and how water levels may impact Phragmites expansion. So how does Phragmites actually survive in high water? We kind of have to start here on the biology of the plant and understand how it's able to persist in high water areas. And it really comes down to the way that the plant breathes, how it gets oxygen. All plants need oxygen, just like every other organism. And so the way that it does this is through convective through flow. And so it's basically transporting gases from above the water to below the water and back out. And kind of the oxygen and air goes through the stomata of the leaves, and it kind of moves back out these dead stems and goes through the plant in a way that oxygenates the rhizosphere, makes the plant happy, and allows it to breathe. It can do this in high water areas too, as long as a portion of that plant is outside of the high water. It's still going to be able to breathe just as effectively. But what happens if the plant breaks? Well, we know that Phragmites can snorkel, and so if, like through wave action, by chance the plants break, as long as there's a portion of that plant still remaining above the surface of the water, it can still breathe in that same way, oxygenate its rhizosphere, remain happy, and regrow. But the whole idea behind cut to drown is, what if we cut off this convective through flow mechanism? We don't allow the air to get into the plant. It's kind of blocked by the water. And then these plants, kind of just drown. They're not able to breathe, and they drown if they can't resprout and break the plane of that water. So this strategy isn't new. We didn't develop this. This is something that's been used throughout the basin for a long time, and is actually really a popular management strategy in Canada, where it's being, they're using this in ways with hand tools, and also in larger areas with these cool trucks or things that can come in and cut large areas of Phragmites. If you would like to hear more information about kind of the strategies that you can use for cut to drown, I'd encourage you to go back. I think this might have been like 3 years ago now, almost, and watch Dr. Janice Gilbert's presentation in the same venue at the GLPC webinar. She gave a great talk, and all of this information is still very relevant today. But it talks all about how to achieve good results using cut to drown. So I mentioned that this work is supported by a USGS and Fish and Wildlife Service Science Support Partnership grant. And we put together this project because we realized that while this is a management strategy that's used kind of widely, there's a lot that we don't know. The literature is kind of devoid of information on best management practices for this strategy. And namely, we came up with these questions that we thought were kind of critical to understanding how well this will work. One is, what is the optimal timing for cutting? Should you cut early in the year or late in the year or multiple times? What are the rhizomes actually inviable after drowning? So if they're drowning, if they drown and they don't resprout, will they ever come back if water levels go down? And what if a clone extends into low water? And what I mean by that is, if you cut a clone of Phragmites, where they're all connected underground, and you cut this patch that's entirely flooded, you may be able to achieve an effective kill. However, if you cut it and the clone extends into a low water area or into a shoreline where it's not flooded, it's likely that those plants may be able to provide oxygen or resources to those flooded stems and allow them to resprout. So we wanted to get a sense for how effective this management strategy may be in these areas where clones may extend into low water, and what we could do as managers to alleviate that. So throughout the study, we're going to show results of a greenhouse experiment and preliminary results from a field experiment. And we're going to kind of follow this strategy for presenting our results. And these are kind of our measures of success throughout. We're going to show how our treatments affected seasonal growth. So in our greenhouse study, this is going to be in the form of below ground biomass. In our field study, this is going to be in the form of stem density. We're also going to look at how these treatments affected growth capital, what I'm calling it. So the ability of these plants to regrow, how much resources they have to be able to put up a new shoot. And to do that, we measured the non-structural carbohydrates in the rhizomes, because these are the sugars that they use to put up new shoots. And then we also just looked at straight up viability. So we cut some rhizomes, took them back to our growth chamber and see if they resprout. So just straight up regrowth, can the plants regrow? So we'll start by talking about this greenhouse study that we did in 2021. And I'm going to turn it over now to Spenser Widin to give us the details of that study.
Thank you, Wes, for that introduction. My name is Spenser Widin, and I'm going to talk about this greenhouse study that we did in the summer of 2021 at the University of Michigan Botanical Gardens. And this study was primarily looking at the first two of those research questions that Wes introduced. So looking at what's the best timing for cutting these plants underwater, whether early in the season, growing season, or late in the growing season. And then also what is the future viability like of these plants following a cut to drown treatment. And so to begin to address those questions, we designed this greenhouse experiment where we actually took some field collected rhizomes, propagated them here at the Great Lakes Science Center in our growth chamber until they were big enough to then transplant into some soil, let those establish in the soil, and then we're able to apply these submergence treatments by stacking them on various numbers of bricks within these cattle tank mesocosms. And through this, we were able to achieve 3 different submergence treatments. Either the plants were not submerged, so at the soil was at the height of the water, plants were either partially submerged, so a portion of their stem was underwater, or plants were totally submerged, so the entirety of that below ground and above ground biomass of that plant was submerged. And then we were able to combine this with 3 different cutting treatments. Either as our controls, plants were uncut, plants were cut early in the season, so late spring, or plants were cut late in the growing season, so late summer. And then as Wes touched on, our measurements of success for this was looking at the below ground biomass as well as the carbohydrate concentration of those plants that we harvested. And then a subset of those plants we actually allowed to overwinter under natural conditions, senesce, and then store overwinter them, and then put them back within our growth chamber under ideal conditions to measure regrowth as a way to determine viability of plants following cut to drown. And so if we then look at our results from this experiment, first looking at our below ground biomass, here you can see in this figure on the y-axis, the vertical axis, we're measuring the total below ground biomass, roots and rhizomes of those pots in grams. And then those are plotted based on submergence treatment and then grouped by the cutting that we applied to them. And so first from this, right off the bat, you can probably notice that within our totally submerged plants, these actually seem to die pretty quickly, and they were not seen to actually produce much of any, if at all, any new below ground biomass. So cutting off that oxygen to those below ground structures seemed to be effective in killing these plants and preventing future growth. Whereas in our cut to drown plants, so those plants that were partially submerged and then either cut early or late in the season, this did seem to be effective in reducing below ground biomass. And particularly here, timing seemed to play a bit of an effect in that the early cut plants showed lower biomass accrual than those late cut plants did. Whereas our just submerged plants uncut didn't seem to have that much of an effect at all on the below ground biomass. And then if we look at our non-submerged plants here, not submerging them and only cutting them, whether early or late, had a little bit of an effect on the below ground biomass, but ultimately those plants still produced a lot of growth underground. And really it was kind of just the same as partially submerging plants. So not submerging them and cutting them seemed to be about the same as just partially submerging those plants. And so this data suggests that in our cut to drown plants, as well as our totally submerged plants, this was an effective strategy in potentially killing those below ground rhizomes. But some further evidence of that comes from our carbohydrate results. And so here I'm going to show you a set of 3 figures. The top's going to be the % sugar of those plants. So those sugars are more readily available carbohydrates for immediate growth of plants and metabolism. Starch is more of a measure of the long-term storage of plants. And then the bottom graph, the total % non-structural carbohydrate, is just the sum of both the sugar and the starch. So if we look at our results of this, you can see here again from our totally submerged plants, whether they were cut or uncut, early or late in the season didn't seem to make a difference. Those plants had really significantly reduced carbohydrate content compared to our control plants. This is in agreement with what we saw with our biomass, so really low levels there as well. And then again, if we look at our cut to drown plants or our partially submerged plants that were then cut early and late in the season, cutting those plants under high water conditions also seemed to reduce the carbohydrate content quite a bit. And here, unlike the biomass results, we didn't really see an effect of the timing of cutting. There was similarly low levels between the early and the late cutting. And then just partially submerging plants didn't seem to have much of an effect on the below ground carbohydrate content, whether it's sugar or starch of these plants. And then our non-cut plants, we did see an effect of cutting timing here in that cutting plants late in the season did show a reduced concentration of both sugars and starches within those plants, whereas uncutting them or cutting them early in the season didn't seem to have much of an effect on the carbohydrate concentration of these plants. So again, with our biomass results being really low in our total submergence and our partially submerged cut plants or our cut to drown plants, these really low levels of carbohydrate concentrations seem to suggest that these plants are not going to be viable the following season. But to further test that, again, we took a subset of our plants that we then overwintered, regrew under ideal conditions, and then measured the mean number of new stems produced per plant per day. Here we omitted the total submerged plants primarily because of limited space within the greenhouse. And then also by the time we set this up, plants were a little too big to be able to maintain total submergence. But nonetheless, we do have some data on our cut to drown plants. And from this, you can see that cut to drown, just like the biomass and the carbohydrate suggested, was effective. Essentially, none of our cut to drown plants, whether it be early or late in the season, produced any new growth. So effectively, those plants were killed by this treatment, whereas just partially submerging outcome plants didn't seem to affect the viability. But all of those plants eventually regrew, along with our just cutting plants as well as our control plants. All eventually did regrow. The rate at which new stems emerged seemed to be affected from this, but ultimately non-cut to drown plants did regrow, whereas our cut to drown plants seemed to be killed by this treatment. So just to quickly summarize all that again, based on the results of this greenhouse experiment, cut to drown, just like managers have seen in the field, does seem to be effective. We killed our plants in this study through the use of a cut to drown management. Cutting timing kind of had some various results between biomass production and carbohydrate content, but ultimately our results sort of suggest that cutting timing of these plants may not matter. Both seem to be effective in killing plants, and that just cutting plants alone, unless you can submerge them, doesn't seem to be a successful treatment, at least as an initial one-off kind of thing. And so, of course, there are some limitations to this study, being that it's a greenhouse experiment, small plants, really controlled setting. And so this kind of begs the question of what about wild populations? What does this mean about wild populations? And so to kind of start to give a little bit more background on the field studies we did to address this, I'm going to pass it back off to Wes to give an introduction to our field work we did last summer.
Thank you, Spencer. Kind of playing musical chairs in here, so bear with us. Thank you. So yeah, so what about wild populations? Obviously we had some really promising results in the greenhouse. Things looked really good. We were killing our plants, but it begs the question, what's going to happen when we take this out in the field and we're not dealing with these tiny plants anymore? We had these limitations with our greenhouse study. One that you can see when we started these plants in those tanks. These are tiny plants. They're growing from one small little rhizome chunk in an isolated kind of limited variability system. So it doesn't, while the results are promising and really cool to suggest that it works, moving to the field was going to be a challenge. They also have much smaller existing carbohydrate reserves than wild plants do. Over many years they're going to build up these reserves and maybe much more difficult to kill. And lastly, this greenhouse study wasn't able to address every research question. I mentioned this question of connection to lower water plants, and we weren't able to address that in our cattle tanks in the greenhouse. And so this is something that we needed a field-based setting to be able to address. Okay, so last summer, we started to address these questions in the field, and after a long search for field locations, we came up with 2 locations that seem to fit our needs very well. One is the Fish Point State Wildlife Area in Saginaw Bay of Michigan, and the next is at Metzger Marsh in Ohio. And I'll talk just a little bit about those 2 field sites. So the Fish Point in Saginaw Bay, this is what we're calling the Garner Road Marsh. That's how we'll refer to it for the rest of the presentation. And this is managed by the Michigan DNR. This is right by the Saginaw Bay, but it is an impounded wetland. So you can kind of see in that figure in the upper left-hand corner, that's actually the lake. So it's really close to the lake, but it's not impacted by water levels in the lake. But as an impounded wetland, the cool thing about this was that it's dominated by Phragmites and cattail, and it also has water control. So the managers there said, well, we can pump a bunch of water on this for you. And so we saw this as an opportunity to kind of do this controlled experiment in a natural setting, where we could set up a bunch of plots throughout the site and ask these more complicated questions, kind of being able to look at connection and also timing of cutting in research plots, in a natural setting. So it was kind of a cool site to work in. The next site we worked in was at Metzger Marsh in Oregon, Ohio. And so this is much different from the Garner Road site. One in that it is a true coastal wetland and it's maintains some connection to Lake Erie. However, it's mostly, it has a water control structure, can be connected to Lake Erie, but it's for the most part closed off. But they maintain high water levels at this site for several years, or they have for several years. This is right adjacent to the Ottawa National Wildlife Refuge, and it's partially managed by the U.S. Fish and Wildlife Service and also the Ohio Division of Wildlife. And at the site, we noticed that there were these kind of like isolated patches of Phragmites throughout. And you see these circles here. These are all little patches of frag that look a lot like the picture down there where you see the person in the canoe, where these are, patches of Phragmites seem to be isolated from the others. They're growing in about 1 meter of water. And they seemed like really perfect locations to test this strategy of cut to drown, because these patches were something we could handle with our tools that we were using, and would really kind of be perfect for testing the different treatment options that we had. So to get into the details of our experimental design, I'm going to turn it over to Kaira Liggett now to talk about what we did at each of these sites.
Okay, yeah, thanks, Wes. So yeah, I'll be talking about the field experiment as well as some of the preliminary results we talked about. So at Garner Road, we had a total of 6 treatments and 5 replicates, and the treatments varied in cut timing and connection type to unflooded frag. For the cut timing, we had a no cut, a continuous cut, which included multiple cuts throughout the season, and then a single late cut in August. For the connection type, we had a disconnected type and a connected. And then again, as Wes mentioned, we were getting at that question about whether partially submerged or cut partially submerged, whether cutting submerged portions of a partially submerged frag stand, whether connectivity to unsubmerged portions, whether that would matter or not. And so for the disconnected plots, we utilized a serrated spade shovel to go around the perimeter, either on 2 sides or all 4 sides of the 3 by 3 meter plots. And that was an attempt to sever those rhizome connections. So at Metzger Marsh, we just had 3 treatments varying in cut timing. So we had no cut, a continuous cut, again, multiple cuts throughout the season, and then a single cut for our late cut. And then at both sites, we did utilize a gas-powered hedge trimmer to apply our cutting treatment. And so then, as you can see on the left, at Garner, we were able to, cut down the frag and then stack it within the Garner Road site. And again, using that hedge trimmer. But at Metzger, it was a little bit different story. So at Metzger, because the patches were more spread out, there's potential for the frag, once we cut it down, to float away in the marsh. So once we cut down our frag patches, we actually had to load it into a boat, which usually took about 3 or 4 people, with a lot of manpower. Once we had the frag loaded into the boat, we would then bring it back to shore, where we would load it into a truck and take it off to a composting center. And so, yeah, that's just something to note, just having a plan for disposal in place if you're applying this cut to drown treatment. And so before we get into the preliminary results, I just wanted to remind you how we were measuring success in the field site. So we were looking at above ground growth, measuring stem density, growth capital, looking at the rhizome, carbohydrate storage, and viability, looking at the regrowth from those rhizomes at the end of the season. So at Garner Road, we saw that cutting did significantly reduce stem density for both the late and the continuous cut plots. And so here, as you can see circled for the continuous cut plots, we did notice that there was some regrowth, but it wasn't many stems that were coming back. It was in the single digits. And then you can see the green control line. That's just, we were measuring, because we were measuring, looking at the live stems for stem density. That's why you see that decline throughout the season as things start to senesce. There's just a decline in that. For Metzger, we saw that something similar where stem density was significantly reduced when compared against the control plots. However, we did see a lot more stem regrowth. So stems were coming back in much higher numbers. You can see this is a different scale representing a lot higher stem density. And so we wanted to take into consideration the fact that the stems between Metzger and Garner, they did look different. So as you can see on the right, the stems at Metzger kind of look like this, where there's a lot of those hairy adventitious roots, when compared to Garner was like most of the stems had little to none of these. And so these roots were growing below the water line, but above the soil line on the stem nodes. When we collected stems from both sides, we saw that Metzger did have significantly more of these roots on their stems, but these roots are thought to aid in oxygen transport. So we thought that this might be maybe a part of the reason that Metzger was a little bit more resistant to our cutting treatments. But bottom line, stem density was reduced at both sites. Additionally, in the case of stem density, the connection type didn't seem to matter, but we are exploring that a little bit more when it comes to some of the below ground data. Next, for growth capital, we did see that the percent starch at the end of the season was reduced significantly for both the late and the continuous cut plots, but especially in the continuous cut plots. We still are waiting on the percent sugar data, but again, as Spenser mentioned previously, that percent starch is like the long-term storage. And then lastly, for the viability of the rhizomes, we did see upon collection at the end of the season, after we'd applied our cutting treatments, that these rhizomes were still viable. So they still had the ability to put up new shoots even after the treatments we had applied. So we did see that the treatments did affect the speed of regrowth, but again, they were viable. So I'm going to hand it back to Wes to wrap everything up.
All right, thanks, Kaira. So what did we learn? We, over the past 2 years, have been looking at this cut to drown strategy in a couple of ways, both in the greenhouse and in the field. And I think the takeaways are that we have proof of concept that this can be extremely effective. That cutting plants underwater does limit oxygen to the plants, does kill them, and at least in the greenhouse, all of our plants died. We saw significant reductions in biomass. We saw the significant reductions in that growth capital and in viability. Moving to the field, I think we have really promising signs that this could be effective. The above ground growth was limited in our cutting treatments. That growth capital showed some signs of reduction over 1 year. And even in the viability, even though these were all viable, we saw that the speed of these plants resprouting was affected by our treatments. But we do have to say, they were all still viable. So if you were to go out and apply just one season of cutting, you wouldn't likely get the results you were hoping for. Because if you didn't do anything else, these plants are just going to come back, and eventually, they will replenish those rhizome stocks and get stronger. One of the reasons that we think that may be occurring, or something that may be affecting it, is the formation of these adventitious roots, especially if they're involved in oxygen uptake. And we have a student working with us who's interested in studying this more, so we may get more information on this topic in the coming years. But all of this really points to the fact that this is likely a multi-year management approach. That in the field, we are not likely to get a total kill after 1 year just applying this cut to drown. And I think this is consistent with what others have found, in their work as well. So the next steps here is that because we're looking at this as like a multi-year management strategy, we'd really like to return to these sites in this summer. And we're working on developing a plan for that and talking to our partners about getting back to the same locations. And then we'd also like to tie this to the PAMF treatment alternatives. And Teresa mentioned the PAMF program, and obviously a lot of information on the PAMF program at the GLPC website if you're interested in more. But one of the things that they developed through PAMF at the very beginning, when they were putting it all together, is these treatment alternatives. And these are supposed to be the kind of all of the treatments that our expert panel could come up with for how Phragmites is treated throughout the region. And you'll notice the one that deals with cut to drown is a cut underwater during the translocation phase, which would be at the end of the season, so like a late cut, as we were calling it. Rest during the dormant phase, so nothing during the winter. And then in the growing season, another cut underwater. So that would be like akin to an early cut. And so we'd like to kind of match our treatments to what PAMF recommends, because we also have some evidence that it could be really successful. So at our Metzger Marsh site, Kaira just showed that the percent starch was really reduced through our treatments. The late cut reduced the percent starch, but that continuous cut, although we were getting stems coming back, because we were cutting them, every week or every 2 weeks, we really reduced the amount of growth capital that they have. They don't have many resources to put up new stems. So if we were able to get out and cut these plants when they look like this, like the picture on the right, when these new shoots have just come over the surface, it's possible these plants have kind of expended all of the resources that they have just to get them above the water. And so cutting them at that time before they're able to replenish any of those resources could be really effective in year 2. So that's what we're hoping to get some data on next summer. So with that, I'd just like to say thank you to a bunch of people. I'd like to say thank you to all of our partners at the U.S. Fish and Wildlife Service, the Michigan DNR and Ohio DNR. The University of Michigan was extremely helpful in our greenhouse study, and also some early help from the Chesterfield Drains Commission and the Lake St. Clair SISMA. You can see a whole list of people on our team and all the people that have helped with the project over the last couple of years. Even if it's a familiar name, you see Teresa's name on here. She was, before she was at the commission, worked with us on this project for a few months, and we're really happy to have her help as well. Also, just like to thank our funders, the U.S. Fish and Wildlife Service and the USGS, and recognize all the PIs on that project. If you'd like to contact me, my information is here. And I think we probably have some time for questions. Thanks.
That was great. And we have a lot of questions. So okay. For you guys too, I also put you guys' contact information up. So type any questions in the chat and we're going to try to address them all. I will just address them to you guys as a team, and then you can decide maybe who wants to answer. Okay. Also, Patrick might be able to help me too. I know he's been sending them to me along the way. Okay, so this is from Abby. Are there particular herbicides that target Phragmites respiration processes that you guys know of?
Not that I know of. No, I mean, the herbicides that are commonly used are systemic and, kill the plant. And so, in effect respiration because they die. But I don't know of anything that could target, how they breathe. That's what the question is asking.
Okay. From Dustin. So if areas cannot be adequately drowned, would the best management practice be drowning the areas that is possible and treating the rest with a chemical pesticide, especially if they're sharing the same rhizome?
That could be an option. Yeah, obviously we did not test that directly, but in our work with, I said early on we talked with some SISMAs, and there were a few that were applying that strategy, that were working on kind of shorelines, and were, herbicide's expensive. So they were looking for a way to reduce the amount that they were using at those sites. And they said, well, maybe we could target the shorelines with the herbicide and cut the rest underwater, the stuff that we could actually drown. And I am not sure, obviously we don't have data on how effective that was, but it seems like if you could kill those plants on the edges, you would at least stop them from being able to give oxygen to the plants that you've drowned. So it seems like that could be an effective strategy potentially.
Great. This is from Laura, and just I think a clarification on your guys' methods. She said, for the methods, either of them, you must remove the tops and have a disposal tactic in place. Is that correct?
Yeah. So this was a much bigger job than we had imagined. And I think all the pictures that Kaira showed were indicative of that. You saw the one boatload of frag, and I was thinking through this morning as I was like looking through the final slides, I'm like, I think there were probably at least 20 of those boatloads of frag that we produced. And, we were just doing these small experimental plots. So if you've got a large expansive area, you really have to have a plan in place. And, it could be that you find a place to pile it up on the site. That would have been ideal. It just didn't exist in the location we were working on, at Metzger Marsh at least. But if not, you have to have kind of a way to, a plan for getting it out of there.
Yeah. This is from Patrick. So do you guys have any idea if this would be effective for sites that only flood periodically? Can you speculate, or are you aware of how long of a duration of flooding after cutting would be needed to be effective? As many sites don't have flood control that, you can put in place.
Yeah, that's a really good question. And I think, the answer is going to be unsatisfying because it's probably site specific, because if you have a really small patch, you may be able to kill it, in a short amount of time of flooding. But if it's a really big, patch with lots of, a long history, meaning that it probably has lots of resources in those rhizomes. We saw, just at Metzger, we were coming back every 2 weeks and recutting, and these were flooded completely, and they were still putting up new shoots for the entire season, basically. So if you, only maintain flooding in a site like that for a small amount of time, I would think those plants would be able to outlast that if they had a really, good base of resources, I guess, that they would probably outlast it. Anything else you guys had on that?
No, I think that, it would be site specific, and, the age of the stand, like you said, would play a part.
Also, these are really great questions, guys, from the chat. So this might be kind of similar. So if you don't have anything to add, that's fair. Would cut to drown method work best with a combination of spraying chemical pesticides as they drop the nutrients for dormancy?
I think like I haven't thought of it in combination with herbicides, like at the same locations. I think that the distinction before was like to get it on the edges with herbicide and then cut to drown. And I think like the hope would be that you wouldn't have to use herbicides, that you could use this instead over multiple years. But it could also be used, as a follow-up to herbicide. I think that, so one consideration, I guess, would be if you cut these plants at one of our sites, we didn't get a lot of regrowth. And so it would kind of be like, what are you spraying at the appropriate time of year? There wouldn't be that many stems to spray. But you may have a site like our Metzger site where you did get a lot of regrowth, and maybe an effective way to kill those would be chemical herbicides. But as our data are suggesting, and I guess we'll know more about this in the next year, it seems like we may have an opportunity to control them without herbicides at all, just by cutting them in that second year if you get them at the right time.
This is from Dan. Did you compare single year effectiveness of the cut to drown method to that observed of maybe a single year herbicide treatment?
We did not, but that's a really good point. And something that, we had such good results with our greenhouse study that we were like, oh, this is great. This is going to, this is going to work so well in the field. And, as we saw, it was, kind of a mixed bag and promising results, but it was regrowing. we were sharing this with our partners, project partners, and at Fish and Wildlife Service, and they're kind of like, well, you know that we always have to retreat. I mean, it's just like, it's not common that you just kill everything. And I think we kind of gave ourselves a false sense of like hope because the greenhouse study went so well. So I imagine, yeah, it would be interesting to look at how effective this strategy is compared to just herbicide in terms of effectiveness in 1 year. So that's something that would be interesting to look at.
This is from Stephen. Was the Metzger site maybe more eutrophic, lower dissolved oxygen, and so that the plants had developed those adventitious roots to help with oxygen exchange?
It's possible. We did not measure dissolved oxygen at these sites, unfortunately. And one of the major differences between the two is that the Metzger Marsh site had been flooded for many years, and so these plants were kind of used to being in high water, and so may have developed those roots as kind of a kind of plastic adaptation to these high water. Whereas at the Garner Road site, it was flooded that spring before we started our cutting. So these plants were not used to being flooded in the same way. And so we did see that at the end of the season, there were some adventitious roots that had formed at the Garner Road site. They were just in much lower numbers. And we know they weren't there before, because why would they be if there was no water? So I think it's cool that there is a student who's interested in looking at this question a little bit more, because I'd like to know that question too. Is it a response to just being in water, a stress to the plants, or does it have something to do with the chemistry of the water, whether it be nutrients or dissolved oxygen or turbidity or something like that?
Yeah, that may be an update webinar. We always love to give those. So
yeah.
This is from Laura. I think you can either share the slide again or just mention it. She said, can you repeat what you mentioned for the most important 3 steps? You circled on the next step slide.
Yeah, so the expert panel, I think we're referring to as the PAMF guidance for treatment alternatives. And the expert panel developed this guidance which for cut to drown recommended a cut underwater during the translocation phase, so like at the same time that you would be spraying, late in the season, and then a rest during the dormant phase, so like right now, and then another cut to drown in the growing phase, so that would be in the early part of the season. And I think that if you maybe contact people at PAMF, they'll tell you, I guess, exactly what that growing season phase is, because I think there are some actual months prescribed to that, and maybe Teresa even knows.
Yeah, Laura, if you have questions, you're welcome to email us about that. This is from Dustin. Do you believe that repeated cut to drown treatments could reduce the growth capital to a point where it's eliminating the rhizomes?
Yeah, I think we have good evidence that, especially when we compare our continuous cut to our late cut, we had, there was a significant difference in the amount of starches present there. We'll see what happened in the sugars, but, it's likely similar. The plants are using those resources to put up new shoots over the whole season. There's at these patches that you saw, there were like hundreds of new shoots that were coming up every 2 weeks when we went out to recut them. And so we're cutting all of those again before it has much time to replenish those. And so, just think about your bank account. If you're spending more to put these shoots up and you're not able to make any more money, eventually you're going to run out. And so that's kind of how I look at it. And I think that, that's why I think that these repeated treatments could be really effective.
Great. This is from Dara. Do you think that turbidity has any impact on the drowning process? Did you track turbidity or water clarity at all?
That was something we really wanted to look at. We had some initial conversations with Janice Gilbert about this when we were getting this going. And she said she thought turbidity mattered and had some evidence from some of her sites that it did. And we tried to have a turbidity treatment in our greenhouse experiment. We thought, oh, we can make some of these tanks turbid and some of these not. it was really hard to do. You know, we were like trying to mix clay particles in the water, but it kept settling out, and it was really hard to make it consistent. And then at our sites, we also didn't find sites that were really different in their turbidity levels, so we weren't able to track that in a meaningful way either. But it is something I wish that we were able to do better. And I, you know, if anybody has any recommendations on how we could do that better, I'd be open to it, especially for this year, if there's something that we could do to see how much that matters.
You're doing great. I feel like this is a lot of questions for our Q&A, but these are great. Okay. Is there a concern that composting the cut material may leave viable seeds that could spread around?
Yeah,
I guess maybe in the transfer or in the composting process. I don't know.
Yeah, that's a good question. I mean, so most of our cutting we did fairly early in the season, although we did have those late cuts. So we did it throughout the season. I'd say half of the time in those continuous cuts, there were no seeds present, but later on there were. I think, yeah, you do have to be, I guess, in the transfer. I mean, we had these things floating in water and then we're putting them in the boat. I mean, I'm sure there were some seeds that were coming down, but, you know, similar to if they were just standing there, and even if they're sprayed, I think sometimes the seeds are still viable. So the seeds, yes, I think can be a concern. We, as far as like the stems resprouting, this, you know, might be what others are thinking. Because we often in our greenhouse will start stems in water, you know, to make new plants to work with. If you just let these things float in the water, they may be likely to resprout. But we did have a group of interns this summer who did a little mini experiment. We don't have any data to share, but they're hoping to put together a blog post for the Frag Collaborative about this, just to look at whether there was likely going to be resprouts from kind of mounding Phragmites. You know, if you have to do cutting, can you just like, you know, stack it, and are you likely to have resprouts from that? The preliminary data from that suggests that there were very little resprouts from those stems if they're stacked, especially if they're stacked in a way where most of it's outside of the water. So at the compost facility too, I mean, you know, they're likely turning it enough that these are really drying out and dying there as well.
That's awesome though. I hope they write that, because we do get a lot of questions from like homeowners and landowners about seed viability. So it'd be good to like point them to something to read. Okay, I think there's only a few more questions. How far underwater was the Phragmites cut? Do you think there's any evidence that cutting deeper underwater would be more effective?
I think that's a great point. And so we tried to get as close to the surface as we could. At the Garner Road wetland, we were able to achieve that really well. At Metzger, it was really difficult to get down. And the reason for that is that these were long established stands of Phragmites that had rhizomes that were growing in the soil, but also some of them that were kind of growing above the soil line, and just kind of in this kind of dense spaghetti network. And also there's some submerged aquatics there that made it difficult to see actually what we were cutting. So when we were getting that down low, we were trying to cut as close to the surface of the sediment as we could, but we didn't at all times hit that mark. And I think that's why some of those the adventitious roots may have mattered, because those stems, although they were cut as low as we could have gotten them, still were kind of present, and they had some of those roots on them. So if those were acting as kind of like a oxygen tank for these plants, you could see why they were able to just keep shooting up new shoots. So I do think that it's better if you could get as low as possible to reduce the ability of these plants to get resources from those roots.
Great. We have a question from Laura, and maybe you can clarify, would burning the cutting be a good option? I'm not sure if he's talking about like the cuttings that you would dispose of or the stems itself, but maybe talk.
Yeah, I mean, if it's burning like the piles, I think that would be an option. If you could get that to burn, I don't see why that wouldn't be a good option to get rid of it.
Yeah. and we just have a second that people would definitely be interested in the seed potential.
Okay.
So just seconding that blog post. Okay, Patrick, I guess you can let me know if I've missed any. I think I've addressed them all.
Yeah, I think that's all the questions that I've recorded.
Okay.
If we missed any, please let me know in the chat.
Yes, you can still add them. I'm just going to do some few follow-up slides. Just to remind you guys all, if you wanted to share this webinar, if you're like, this is great, you want to re-listen to it, you want to share it out with colleagues, our webinars are recorded. We will send a recording out to your Eventbrite registration, as well as we'll post it on our YouTube site and our own website. So this information is out there now forever. And also just a pitch that if you want to present, if you have research or work you'd like to present, or you have a Phragmites topic that you would like to hear more about, we always want to hear, what you, the audience, want to learn about. So feel free to email us at thephragmites@glc.org, and we will kind of, get some ideas of what 2023 should look like as far as our webinar. But that's it. So thank you guys.